Right. Next step then is let's make a start on the proteins bit. You know, if you were ever at a loose end and wanted to study something biological, proteins would be a very good place to start. You can't look at very many aspects of biology without having proteins stuck in there somewhere. I mean, looking at this drug design module that they want on the Yarmouth Foundation degree, one of the learning outcomes is to understand the relationship between drugs and receptors and enzymes, although I would sort of run those together and say proteins, because drugs are almost always targeting proteins. Not all of them, but a lot of them. Proteins are entirely what genetics is about, and a huge amount of current research is going on into the parts of the genome, DNA, that don't actually give us the recipe for a protein, in other words, genes, but what does the rest of it do in producing proteins? So, you know, the whole lot is, you know, biology, you know, it's very, very hard to find any aspect of biology that doesn't involve proteins somewhere. So, what are proteins built from? Well, their units are amino acids. They are polymers, the proteins. So, a protein molecule is a polymer, and the monomer units are the amino acids. Now, amino acids are not just found in proteins. I believe there's something like probably about 1,500 different amino acids. I can't remember where I got that number from, but it just floats around in my head, so it could be wrong. But we find about 20 of them in proteins. All right, so it's about 20 different amino acids that are found in proteins. As far as human beings are concerned, we can make some of those 20, but others we actually have to get from our food. They are known as essential amino acids, and it sort of determines what sort of proteins we need to eat. If you eat animal products, then you will get all your essential amino acids without any problem. If you have a vegetarian or vegan diet, you have to mix and match your plant proteins in order to get enough of the essential amino acids. So it's usually a sort of random thing, you know, but things like grains and pulses and other vegetables and that sort of thing, nuts, you know, but a variety will cover them all. Just in a omnivorous human diet, eating everything, what do you think would be the best source of protein to give you the best match of amino acids for the human body? What source of protein? Red meat, you think? No, red meat has just got the protein that you'd find in muscle, hasn't it? Something, what one item of food would have, what is the nearest thing to a whole living organism that we eat? An egg. Yeah, eggs, yeah, sorry. Yeah, eggs are essentially the raw materials for making a whole chicken. Yeah, all right, we're not chickens, you know, we haven't exactly got feathers, but, you know, the component parts of a human being are not that different. There is blood, there is skin, there is muscle, there is, you know, all the different tissues. So an egg contains all the amino acids that are needed for building all the tissues. So egg protein is actually the nearest match to what the human body needs. Meat is a good source, yes, but, you know, it's not as... Meat, if you looked at a sort of percentage match with what the human body needs, eggs come in at over 90%, meat would be in the 80s as a match for what the body needs. So, yeah, you've got... No, uncooked eggs are nasty. Uncooked eggs are... Well, the proteins are denatured, but that doesn't do anything to the amino acids. The amino acids are still there. So we'd still digest them down into the amino acids would be there. So yeah, no, we don't have to. I don't have to leave them raw. No, cooking. There's no benefit, as far as I'm aware, from eating raw eggs, apart from that little bit of risk in your life that you might get salmonella or various other things. Yeah, the only thing that happens to proteins when you heat them up. Now, this is something we'll come on to when we do enzymes, but it's relevant to any sort of protein. Proteins are such complex shapes that if you put a lot of heat energy into the molecule, it will break a lot of the bonds that hold that molecule in its shape and denature it, so it distorts the shape. So you think, for example, you know, egg white, uncooked and cooked are very different, but that's only in the arrangement, the shape of the molecule. It's not, the amino acids in there are still the same. It's just it would take perhaps slightly more digestion, I would think. Well, maybe not. No, I don't know. No, I can't justify that statement. But the amino acids generally then, the 20 of them that you find in proteins, you'll see there is a table opposite the starting page there. Table 5.9, common amino acids in proteins. Yeah, we'll come back to that in a minute. Yes, you need to memorise them all. No, you don't. What we need to start with is our diagram of an amino acid. Right, you have several important parts to it. Now, you will see, obviously, examples of amino acids with a lot more detail than this. This is a generalized formula. We've got three important parts. This is the amino group. So NH2 is the amino group. Now, when you join amino acids together, there's a reaction between the amino group of one and you can see a carboxyl group over there. So they're going to react together. And you always end up in a protein with an amino on one end and a carboxyl on the other end. And I'm trying to think of an example of where we've seen that, and that's all right. I think I know where we saw it, but I won't probably have mentioned it. So this is our COOH carboxyl group. Or that's what makes it an acid, in other words. Woo, not it. So we now know why it's an amino acid. Those are the two bits that determine that. The carbon and the hydrogen in the middle are constant, but what the heck is R? R is not an element. R is an R group, but the R group, R just represents a variable group. And it's the most important part because it's what determines, it's the identity, if you like, of the amino acid. They've all got an amino group, they've all got carboxyl group, those are tiny. What we've got there is tiny. The R group can be huge. So this is the bit that actually determines which amino acid we're looking at. All right, so we'll look at some examples of these and just sort of get a start on this, and then we might, I don't know if we'll get on to joining them together, but identifying some of the variable groups is quite useful. So if you have a look at that table, table 5.9, the amino acids are listed on there by name, by three-letter abbreviation, and those are standard abbreviations. So those are the abbreviations that are always used, those three-letter abbreviations. You'll find when we get onto genetics or protein synthesis possibly that they're sometimes known by a single letter as well. But yeah, that's a story for another day. We've got the R group in the next column, and then a few notes for some of them down the right-hand side. So they're listed alphabetically, which is, well, makes as much sense as any other listing system, I suppose. And there is a common ending to a lot of them. Ene, yeah, so the I-N-E, not surprisingly, is amino. It's that ene in amino. So alanine, arginine, asparagine, and then you have odd ones sneak in like aspartic acid. Cysteine, glutamine, glutamic acid, which is different. Glycine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, proline, serine, threonine, tryptophan, completely different one. Tyrosine, valine, all right? So not difficult words particularly. And a lot of sort of, as I say, common endings there. The three-letter abbreviations are very often the first three letters, but not always, because sometimes you've got common beginnings like glutamic acid and glutamine. So glutamine is GLN, whereas glutamic acid is GLU. The R groups, hopefully these are reasonably clear now. I think I did a fresh copy for this year. The little numbers have, over the years they've faded, so I had to do a new one. But yeah, if you have a look, those are the R groups that are stuck in that space on that molecule. So what's the smallest thing you could have in there? No, H. Right, which one's that? Glycine. Glycine is the smallest amino acid there is because its R group is simply an H. The next biggest one is the one at the top there, CH3, alanine. Because if you think about it, what do you know about organic chemistry? What would be the simplest thing you could put on there would be an H, and yes, that is glycine. The next simplest would be a carbon with its 3 hydrogens, so that's your alanine. So they are very small, but then you look at some of the others and they are huge. You've only got to come down, well, the next one down, arginine. You've got 3 CH2 groups, an NH, 3 NH, a lot of amino in that one, a lot of nitrogen in there. But that, you can imagine, that's going to come out of this structure here, a bit like a Christmas tree, isn't it? Look further down, you've got some with rings in. Histidine's a complicated one. You've got a CH2 group, but you've also got a 5-item ring on there. Further down, phenylalanine's got a 6-carbon ring on it. And then you get down to tryptophan, and look at the size of that thing. And that is attached to this. So you can see in molecules like that, these bits, the amino group and the carboxyl group, are almost irrelevant. They don't really, all they do is help join them together. The bits that are important are the bits that are going to be sticking out, the R groups. There are a couple of others just to flag up on here, that there are 2 which contain something in addition. What I haven't put on here is the elements that we've got: carbon, hydrogen, oxygen, nitrogen, and 2 of them contain, which is, and sometimes you'll get S. Right, there are only 2 amino acids that have got sulfur in. If you look down the right hand side, you can see cysteine, and methionine, So cysteine and methionine both have sulfur in. Cysteine particularly, I think, is associated with egg protein. Now, sulfur, eggs, what's that saying to you? What's the smell? Hydrogen sulfide, bad egg smell. Yeah, that's where it comes from. Egg, well, eggy whatevers. But eggy gas is the it's the sulfur. It's from the sulfur and cysteine. Yeah. So useful bits of information you never knew you needed. But those are the only 2 amino acids that got sulfur in. Cysteine's not a very big one, is it? It's got a CH2 group and then SH sticking off. Methionine is often one that it's the first bit of code in a gene. You get a code for methionine. It's often a start amino acid and doesn't always end up actually in the protein. You've got some other quite weird ones. Proline's a weird looking one. It sort of forms into a ring. You've got a joining up process, sort of structure there, where you've got your amino group and your carboxyl group have sort of reacted with themselves. It sort of turned itself into a ring there. But you can see also down the side there, it identifies which ones are essential amino acids, which ones, those are the ones that we can't make. Therefore, we have to get them from food. So, if you're going to draw an amino acid, well, it would be fairly easy to draw glycine. A diagram of glycine would just very simply have an H So you'd have your NH2 group, and then that. All right, so instead of the R, we've got a hydrogen. You know, on the other hand, and I'm going to wish I hadn't done this, aren't I? If you wanted to put something else on, let's choose the one at the bottom, valine. It may or may not work, but we've still got our NH2 group. We've still got that bit. Right, that's all common to all the amino acids. But then we've got to attach a carbon. I've given it a long bond to make sure it fits in. The carbon with a hydrogen, and two CH3 groups. It's got an aerial. All right, so you can see, you know, just with that one, how the R group starts to become a lot more significant than the rest of the molecule. And yeah, you can see then with some of the bigger ones that, you know, that's going to be the overwhelming sort of component. Now, the reason I'm sort of harping on about this is because when we come to actually put them together to make a protein, it's going to be important. It has. I think that probably can't be right, can it? Yeah, it does say a CH3 and a CH2. You're absolutely right on there.
That's one of the C's.
No, I just assumed it would have to be two CH3s. So yeah, it could be. Well, it's not something I'm going to test you on. So we can perhaps look that one up and just check. But yeah, that's the sort of effect you get anyway. All right, so that's the basic structure of an amino acid. We're going to stop at that point and take a break, and then we'll come back to next job then is to look at the diagram on the handout sheet there. You've got that, obviously the first initial amino acid, the important bits of it. We've looked at the table for some examples of R groups. We've now got a diagram. We should be getting familiar with this style of diagram where you've got two amino acids being joined together, again by condensation or potentially broken apart by hydrolysis. I haven't put on this one minus H2O for condensation, but it will be, and hydrolysis would be plus H2O. And we're going to get a reaction between the carboxyl group on the left-hand amino acid and the amino group on the right-hand one, in this case. And we're going to lose the hydroxyl group off the OH, I should say, the OH part of the carboxyl group off the left-hand, and one of the hydrogens off the amino group. So we end up, again, removing two hydrogens and an oxygen. So it's exactly the same sort of process. So it's condensation again, but this time the sort of bond that's produced is a peptide bond. So we had, for the carbohydrates, we had glycosidic bonds. In the lipids, we had the ester bonds. And in the proteins, we've got peptide bonds, but they're all formed the same way, all right, by the removal of enough atoms to make water. So again, you really ought to be able to sort of just fill in the bonds with this without too much trouble, I think. So we've got over on the left-hand side, amino. You could identify the R group on there as R1 because these obviously don't have to be the same on both of them. There's our carboxyl group on there. I've not left myself a lot of space on the other side, but if we have amino and carboxyl groups and an R2 there next to each other, then we can see hopefully where the reaction, and it's a very obvious one in this one, perhaps more obvious than the last two really, that we're just going to lose these. All right, so we've got enough atoms to make water. So if we remove that, minus H2O there gives us the condensation part of it. So starting with two monomers, we're ending up with a dimer. I'll put the other bit in here because it's just easier than coming back to it. So we can break it apart by putting that back in. Two monomers, condensation, we get a dimer. So what we're going to get as a result of this, is a molecule that we would just generally describe as a dipeptide. Right, a dipeptide is two amino acids joined together. Obviously, you can then have tripeptides, pentapeptides. Now, I used to be able to say to people, oh, where have you heard of pentapeptides? But the nature of the cosmetics industry is that that advert is no longer on television. But it used to be one of the, I think it was one of the Olay products that had, oh, contains pentapeptides, you know. One of the anti-aging ones, yes. So you know, and oh, they've got this in, they've got that in, is always supposed to impress you, isn't it? Oh, you know, big science, but it's not. It's pseudo-science, isn't it? Oh, it contains pentapeptides. So, yeah, I mean, you know, having 5 amino acids joined together and slapping that on your skin. Well, why is that going to be good? It's like this. Oh, I could rant about this for hours, you know. All right. Ah, yeah. I mean, you know, you cringe sometimes at it is this pseudoscience stuff and lack of lack of basic understanding sometimes. But yes, so if we create a dipeptide out of this, then you know, hopefully you're one step ahead of me and you've joined everything together. We've got that much left on that side. We've removed the OH group, so we've got an unpaired electron that was part of the pair in here, joining the carbon to the oxygen. So that's now waiting to be partnered up. And so we create a partnership with the nitrogen over there that has also got an unpaired electron as a result of losing one of the hydrogens. It's kept that one, but lost the other one. And the rest of it should stay the same. And so the label to the peptide bond there, I've drawn it rather heavily, all right, but it's this thing here is peptide bonds are no stronger or bigger or chunkier than any other bonds necessarily. But the dipeptide there still has an amino group on one end, a carboxyl group on the other. So therefore, you can very easily add another amino acid to either end, another condensation reaction, another peptide bond formed. The formation of formation of peptide bonds we will come back to when we're looking at protein synthesis, because we're going to go from I think from proteins. I think the next thing we go on to is the nucleic acids. And once we've done nucleic acids, we'll do protein synthesis. So I put those together because obviously it all links up. So tripeptide through to polypeptide. And what we generally say is that a gene in the DNA is the instructions or the code for producing a polypeptide. Not necessarily an actual protein, but what a gene is in DNA, it's a sequence of chemicals that say, put this amino acid in first, now put that one in, now put that one in, now put this one in again, and then that one in again, and then this one again, and then another one, and so on, until you've got a string of amino acids. But that's not a protein, it's a polypeptide. A protein is more complex, it will twist and it will fold, and it might have other things attached to it. A good example being hemoglobin that's got iron in it. Yeah, and that doesn't come from the genes, that's another part of the processing. So a gene is the code for producing, or the instructions, if you like, for producing a polypeptide and determining what sequence of amino acids you get. All right, so, I mean, that obviously, you know, that's yet to come. But if you look at the structure of proteins, then we do obviously have to start with, you know, what is the sequence of amino acids? And we describe that as the primary structure. But you'll see the next thing that we're coming on to, we've got primary structure, secondary structure, and then over the page, tertiary and quaternary structures. And I'm only going to go through these very briefly because there's a good diagram sheet. Found a nice diagram to go with it, showing you the details there. And then the PowerPoint's going to give us some examples as well, because I do like to show you, you know, various different shapes of molecules and illustrate it. So primary structure is quite simply, you know, if you were to go through some amino acids off this list, right, primary structure of a protein is glycine, histidine, asparagine, histidine, proline, phenylalanine, histidine, glycine, tyrosine, valine, 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 serine, proline, and that would be the primary structure. You know, it's reading your protein. And that is one of the really important things in biology these days, is getting protein sequencing. Actually determining what order the amino acids come in, because once you've got that, you can actually reverse engineer then to the gene that must have produced it. So if you know what the protein is, and you know the amino acids in the protein, you can hunt down the gene, because you know the DNA code that must produce that protein. If it was that simple, it'd be great, wouldn't it? Because it's not. But just going to sort of very quickly describe these. The secondary structure is one of two types. There are two sorts of secondary structure to proteins. That once you've got this long string, this polypeptide, which is the primary structure, then on the back of that table 5.9, you can see there's 1 and 2. We have the option of the alpha helix. An alpha helix I've described as being flexible and elastic. A common example is the fibrous protein keratin, which you find in hair, feathers, nails and beaks. You know it's fibrous. And that's the alpha helix nature of that. And the alpha helix, I tried to draw one yesterday for amylose, I think. So, yeah, the diagram sheet opposite does this a lot better. But the alpha helix, you know, if you want to draw one in, well, there's one. I can't draw the next bit, though. But this is another of my favourite biological terms. I don't know if you can remember the first one. It was carbaminohemoglobin. This is another one, which is the beta-pleated sheet. You'll start to see a pattern here. But the beta-pleated sheet is where you get, and you can see it illustrated on the diagram sheet there, but done rather like folded paper, really. It's more like it's not sharp folds. It's more like corrugated iron or plastic or whatever. You know, layers and layers of folded strands, if you like. So you know, you might get here's one layer, and they might be joined together by hydrogen bonds within that layer to hold them as a sheet. And then you'll probably get another layer above that. So this is a separate layer. All right, it's always one of the most difficult things to draw. But it makes for a very smooth sort of protein. And an example of a protein that has the beta-pleated sheet structure and nothing else, right, it's got the primary, obviously, but then secondary structure, it's folded into these sheets, is the protein that you find in silk, which is called fibroin. And it's, you know what silk is like, very, very smooth. You know, that is one of its characteristics, isn't it, the smoothness of it. And it's because, and I think I've got an image of it on the PowerPoint, I think it's just alternating alanine and glycine. So they're very small amino acids anyway, and they literally just sort of alternate. So you've got a very, very smooth structure there. So some proteins will stop at secondary structure. So keratin and fibroin are examples of that. However, others go further and can be absolutely huge. You know, we're talking hundreds. I'm not sure if we get into thousands of amino acids, but definitely hundreds. And what we then move on to is the tertiary structure. And tertiary structure of proteins is one of the most important aspects of proteins. All right, so I've said proteins themselves are important, but tertiary structure. We have already met applications of this because it's the tertiary structure that creates stereospecificity of, well, you can go right back to the first unit in cells, so things like the transport proteins, like the active transport pumps for sodium and potassium. They're shaped like that because of the tertiary structure of the protein. They are proteins, and they are shaped to take sodium ions and potassium ions, and that's because of their tertiary structure. If they form wrong, they don't work. That's one of the things that happens in cystic fibrosis. There's a transport protein in the membrane that moves salt, essentially, which then causes the movement of water. And the protein doesn't form properly because of a gene mutation. And so it doesn't pump salt. So water doesn't move. So you end up with thick, sticky mucus on internal membranes. So the shape of these proteins is critical, and that's the tertiary structure. Now, tertiary structure contains both of these. So we'll see diagrams of tertiary structure that have got spiral bits and foldy bits and spiral bits and foldy bits and a few straight connecting bits and then more spirally bits and another spirally bit and a foldy bit. And that's what tertiary structure consists of. But if you've got a specific primary structure, you've got a sequence of amino acids, you know, that goes A, B, C, D, E, F, G, they will always have the same R group sticking off. Right? Now, you know enough about, you know, interactions between atoms to know that, well, if you've got these groups sticking off, you're going to get things like hydrogen bonds, for example. You get some polar bits and some non-polar bits, and you get those odd sulfurs as well. And this creates interactions which fold the whole thing up. And you get this tertiary structure, it's sometimes described as being 3D or globular. That's a lovely word, right? Globular proteins rather than fibrous. You know, they have this three-dimensional structure, and it is always the same for a given protein because it always has the same amino acids, it always has the same R groups, so you'll always get the same interactions, so you always get the same shape. You've only got to have one of those amino acids go wrong, you don't get the same R groups, you don't get the same interactions, you don't get the same shape. And that's what happens in cystic fibrosis and a lot of other genetic conditions where you get mutations in the genes, you get the wrong sequence of amino acids. So the protein doesn't fold up right, so it doesn't do its job. It's as simple as that. You know, that's a genetic disease. That's what happens in a lot of cases. But that's where we're going with this. The way I've designed this unit is so that this bit on proteins will then lead into DNA, the genetic code for proteins, not for characteristics. That's, you know, that's for the other unit, but for looking at the basics of protein production and then what happens in a mutation to the protein that's produced. So hopefully that the idea is good. Right? The design makes sense in my head. We'll see when we get there. So this idea of tertiary structure, this is the key to receptors, to channels, to enzymes, antibodies, remember? They have to have the specific shape, don't they? Here's my antibody impression again. And yeah, you know, the tertiary structure will come back to you many, many times, I'm sure. You've then got quaternary structure, the fourth level of protein structure. This is where you get more than one polypeptide chain. It could just be 2. It could be 4. Right? But that's not linked to the quat bit of quaternary. Quaternary just means fourth. But I used the example of 4 because that's hemoglobin. And with hemoglobin, you also have other stuff attached as well. So there is a big gap on the page. You know, if you feel like drawing a sketch of hemoglobin, then it looks something like this, where you have a couple of, those are alpha chains. And then you have 2 beta chains. The polypeptide sequence, the amino acid sequence is slightly different in the alpha and the beta. But in the middle of each one, you've got a heme group with iron in it. All right, we had, you know, we talked about this when we were doing blood, didn't we? And the fact that the oxygen binds to the iron in the heme groups. So that protein has quaternary structure, more than one polypeptide, plus other stuff as well. Right, now, easiest way to progress from here is for me to find this PowerPoint. And be in there. With a bit of luck, I've put an updated version of it in, not there, we're now in there, aren't we? Protein structure, yes. All right, you have the printed off version of this with the little notes bits by the side. I'll make sure that this PowerPoint goes onto Moodle so that you've got a bigger version of it, because some of the detail on here is obviously going to be too small to see. But if you know, you'll have the, or you can use the narrated version when it gets onto YouTube. Okay, now the first slide on here is really just, in fact I've put 2 on, that are really just other versions. In fact, I think it is actually the one that you've got. I think I did that on purpose, but I can't remember. Let's say I did. So the first slide is really just recapping those 4 levels of structure that you've already got. So the primary structure at the top there is literally that sequence in the chain of amino acids that you could just read or write down. You could decode it. You could take a protein, you could chemically break it into chunks and work out what that sequence is. The secondary protein structure, you've got the pleated sheet or the alpha helix. So yeah, it's a slightly different version, isn't it? So the alpha helix is the twisted corkscrew sort of effect, pleated sheet folded. And then within tertiary structure on that one, you've got, which isn't on yours actually, it's interesting, it's the same diagrams but not labelled in the same way. You've got on the one on the PowerPoint is actually pointing out there are bits of pleated sheet and there are bits of alpha helix. But I'll show you some visual sort of models of proteins that make that a lot clearer. And then the quaternary structure where you've got more than one amino acid and you would possibly have other stuff on there as well. Okay, a colourful version of it makes it a little bit clearer, particularly when you start looking at your primary structure there. If you're taking the left-hand blue bit here, that's showing a bit of pleated sheet. And within that, and it will be far too small to see unless you've got some very strange vision, you've got the amino acids actually laid out in there. Similarly for the alpha helix on the right-hand side there, sort of showing how that shape is stabilized by the structure of the individual amino acids. But then the tertiary structure, you've got this bit here showing you there's a bit of pleated sheet and there's a bit of alpha helix. So a larger molecule has got bits of the smaller scale structure in there, and then the quaternary structure down at the bottom, scarily coloured. Not something we're going to need to think about so much now because we've done haemoglobin. But the tertiary structure bit we will definitely come back to. And primary structure when we're talking about protein synthesis as well, obviously. Now, the next bit, this is a new addition to my slides this year, because I wanted to show examples of each level of structure. I thought, well, I'll choose a protein that we have met already, and so there's insulin. And it shows you insulin's not a particularly big protein. All right, we've got 2 polypeptide chains in insulin, and this is literally just giving us the primary structure and not saying anything about the shape of it, apart from interactions between some of the amino acids. But you've got the A chain and the B chain. Again, that might be alpha and beta in some sources of information. The A chain made of 21 amino acids, the B chain made of 30. And one of the reasons for me putting this up is that I want to start you off thinking in the long term about the presentation that you do at the end of the last biology unit. Now, one thing that is sometimes chosen is production of human insulin, which is not, I don't mean in people, I mean artificially. So for people with diabetes, how do you produce insulin? And it's created by genetically modifying bacteria to do it. Okay, so you put into bacteria the human insulin gene, and then they churn out the protein. All right, so that is it's a really good topic because it's basically on applied genetics or DNA technology, that presentation. All right, so you could usually if people choose to do that one, if somebody, because only one person can choose each topic, if somebody chooses to do that, then they'll talk about the structure of the insulin molecule to start with, because you can't really talk about how it's going to be created if you haven't said something about what it is first. And you can see the one thing that's highlighted on here specifically is links between these esters, which are sulfur, because you can see they're links between cysteines. All right, so cysteine to cysteine, what they call a disulfide bridge. We'll meet those again in a bit. And again, between the 2 chains, so we've got between the 7 up here and the 7 down there, another disulfide bridge, and then between the other 2 over on the right-hand side, another disulfide bridge. So that sort of stabilizes the structure of those. So that's you know, we know that insulin has this sequence of amino acids. The secondary structure diagrams, it's quite hard. Not actually sure, as we go through, we might not be able to see all of the detail I want. The secondary structure, though, this is our keratin structure. So the alpha helix idea, it's the, you know, this twisting shape, the spiral shape. And if you were to, you know, you look at that on there, you can see where the nitrogens and, well, you can see the different parts. You can see peptide bonds, for example, between a nitrogen and a carbon that's bonded to an oxygen there. So these are our peptide bonds in there. Probably, yeah, it's a bit hard to go all the way through them. So you're getting this spiraling sort of effect, and that will be stabilized by hydrogen bonds. All right, here's one. Here's one. Between hydrogen and oxygen components on there. The helix is actually kept in shape by hydrogen bonding between the different amino acids. And sometimes you'll see that sort of a model, if you like. Then you've got the one on the left here, which is rather as though you'd built it yourself out of the molymods. Now, the beta-pleated sheet, on the other hand, it is just alanine and glycine on here. So you've got, this is two layers. So you've got an upper layer and a lower layer. And so it goes alanine, glycine, alanine, glycine, alanine, glycine, and then same along here, alanine, glycine, missed a bit, I think, alanine, I'm not sure now, glycine. And then similarly on the lower one, where you've got your glycine, alanine, and then underneath glycine, alanine. And that's all there is in that particular protein. So this fibroin, the beta-pleated sheet arrangement, you can see, you know, that why that's going to be a lot smoother in its sort of texture there. All right, so that's primary and secondary structure. Now, it gets a bit more complex after this. The tertiary structure, this is a little section. I did wonder whether to use a different diagram for this, but this one actually works okay. This shows some of the forces that actually keep the tertiary structure, you know, keep the shape, or maintain the tertiary structure. And you have all sorts of things. The ones that we will understand best, well, you've got a hydrogen bond here. All right, so that we're familiar with the idea of hydrogen having a slightly positive charge in that arrangement, and the oxygen likely to have a negative. You know, it's going to be more electronegative, so it'll pull the electrons towards it, so we get a hydrogen bond. We do occasionally get ionic bonds, which you think of as, you know, being really strong forces between ions, you know, where electrons have been transferred. You actually get them within this molecule. What we've had here is a hydrogen, which was on here, has been transferred across to an amino group, and we've actually got a charge set up there, so that the bonding between those parts will be very strong, hard to break that. We've got, I'm not entirely sure what the hydrophobic interaction is here. I mean, hydrophobic, these are the non-polar bits. But they will interact with each other. I mean, non-polar will dissolve non-polar, won't it? If you think about it, you know, you use non-polar solvents for removing oil off clothes and things like that. So that's this one. That's a little bit more sort of off the track of things we've done. And then this thing, the disulfide one that I was talking about. So we've either got cysteine or methionine giving us those sulfurs, and we get this disulfide bond or disulfide bridge between them. So the tertiary structure has got an awful lot going on. It's got a lot of forces that actually hold it in shape. Now, of course, if you change the sequence of amino acids, you're not going to get those, are you? You're not going to get those same interactions. You know, if we take this one out, if we change that for something else that doesn't have its oxygen sticking up there like that, that's not going to happen. That hydrogen bond's not going to happen, and the shape of the molecule will change. So you've got to have, you know, you'll always have the same shape for the same primary sequence. Now, some examples of these then. I'm just going to show you some different example molecules, really. I don't think this one's got a name, particularly. But just to show you different modelling styles. Let's just see if I can park that. The situation with this one is you would start at the bottom end. Now, what you've got down here, that is an amino group. It says NH3+, all right? Although I know it says plus H3N, but that's just written around the other way, so it's actually it's an NH2 group with an extra hydrogen that makes it positive. But it's written that way round because it's the nitrogen that's attached here. All right, so it's just written backwards. So that's our amino group, but it's gained a hydrogen. Well, where's it gained it from? Well, actually, that's up here. That was our COOH. Well, it's the OOH group, but it's lost its hydrogen. All right, so what we had was COOH. That went down here, joined the NH2, and left that negative up there. So that's why that says O2 minus. All right, so you just have to think about what you know and then work it out. All right, but this is the amino end, so it'll go, you've then got a chain all the way through here. There's a lot of amino acids in this. The cylinders, which have letters on them, so you've got A, B, C, D, E, and F, are bits of alpha helix, I think. And then the arrows are bits of beta-pleated sheet, and the green bits are just connections. They're bits that don't actually have any specific form. Now, where over here it says CAMP, that's cyclic AMP, we have met that. That was in the peptide hormones. It's a second messenger. I'm not sure why it's been written there. I mean, that's not what this is. Cyclic AMP isn't a protein. But it may be that that's where that binds to do a particular job. So yeah, I've just sort of I've picked images that I haven't added any explanation to. So just to show you different types of modelling. So there's one with cylinders and arrows. Here's one which is literally just, oh, that's a disgusting pair of colours. You've got, there's fairly obvious alpha helix in there, but there's also some arrows. I don't know if you can see them. There's one easy one to see is up here. Right, there's an arrow. So that'll be beta-pleated sheet, and then obviously the coiled bits are alpha helix. I'm not sure why it's two colours particularly. I'm just wondering, I was thinking to myself, I haven't seen this before, and I haven't. This is a new image for this year. Beta-lactamase. Now, beta-lactamase is an enzyme that bacteria may have. If I get this right, I'll be doing well. I knew at the time when I put it on the PowerPoint, and it tends to sort of then disappear in with everything else. Beta-lactams, I think penicillin is a beta-lactam. Some bacteria can break them down, break down the penicillin. I think it's part of the resistance thing, and I would have to check that. I won't say any more about it. But to do with bacteria and bacteria and penicillin. All right. So just if you want to put a note at the side of that, check out what's beta-lactamase got to do with bacteria and penicillin. Something. Another enzyme. I put that one on because I like the colours. I thought it was pretty. Again, you've got the coiled bits of the alpha helix. The arrows are beta-pleated sheet, and it's also got something else attached to it there. Now, this is an interesting one to show something about enzymes, really. The name of this enzyme, it's D-allo-D-allo-ligase, and you're thinking, yes, so do I know what that means? No, but if you look at it, you might. Alla. What do you think alla is? We've just done it this morning. It was your first amino acid, wasn't it? Alanine? Yeah, alla is the abbreviation for alanine. Now, D it's like the alpha and beta glucose. It's one of the isomers of it. So D-alla, D-alla. All right, that means two molecules of alanine. Ligase. Ase means enzymes. Ligase. What might that sort of enzyme do? What does a sorry? Yeah, I think bring them together. Yeah, like a ligament joins things together. Yeah. So D-ala-ligase is an enzyme that joins together two molecules of alanine. Its name is telling us exactly what it is. Now, alanine, one of the tiniest little amino acids, yeah? So you've got two tiny little amino acids, and to join them together, you need that. Huge. We're talking hundreds and hundreds of amino acids in that enzyme, in that protein, just to join together two molecules of alanine. So it's just a good example of how enzymes are huge in relation to their substrates, is the word that we'll use. I don't know whether those coloured bits in the middle of it are the alanine molecules. Could be. Looking at quaternary structure, there's a much nicer haemoglobin than the one I scribbled on the board. And, you can see, well, what it doesn't do is to show you that there are two different types of polypeptide chain there, because it's actually got them in 4 different colours. But, you get the idea. There are 4 chains and 4 heme groups there. Now, I finished this off with a couple of other images of haemoglobin, just to show you different types of modelling. Whether these will show, oh, they are reasonably clear, actually. This, you can see the top left one here. This is the, the sort of skeleton type modelling that we've been using as an abbreviation for a lot of our molecules. And the bottom right one is called a space filling model. And they are both, well, all 3 of those, that one and these 2 are all representations of the same molecule. All right, so that takes us through these different levels of structure. Okay, right, this all fits together quite nicely. Now, the only other bit that we're left with really is just several tables here of classification of proteins. I'm not going to spend very long on these at all. Really just to flag up, that they can be classified in different ways, and it's really about function. And of course, this, the learning outcomes about structure and function. Because what I want us to do is to have some time to have a look at some of the exam questions, because I need you to get good at those. So you've got table 511 is classifying them just according to structure in the simplest way. They could be fibrous, they could be globular, or they could be intermediate. And it gives you some examples of both. So the fibrous ones, for example, the ones in muscle, for example, myosin, keratin in skin we had. The second table, 512, according to composition this time. And you can have simple proteins or conjugated proteins, which means they have other things attached. The simple ones, as examples, albumins, globulins. We met globulins. And you can see at the side, antibodies in blood. As an example, when we did blood plasma, globulins was there. Histones and scleroproteins, keratin can be put in there if you want to classify by composition. Conjugated proteins have stuff added to them. Now, the bits that are added are called prosthetic groups. So you can have things like phosphoproteins with phosphoric acid, glycoproteins have got carbohydrate, and so on. All sorts of things added to them. So you have, let's see if we can pick one, have we met one before? Lipoproteins in membranes, chromoproteins. Chromo means colour. So chromoproteins have pigment attached, and haemoglobin, for example. We've just had that one. So you've got the globin part is the protein, and then the heme bit is the iron-containing pigment group that makes it red. So you can classify them that way. Alternatively, and possibly a little more usefully in terms of, the just relating structure to function, protein classification according to function for table 513. And so we've got structural proteins. We've got enzymes, a lot more to be done on those. Hormones, well, we've met some of them. So insulin and glucagon are there as examples, so that's good. Transport proteins, we've met hemoglobin and myoglobin. Protective proteins, we've met antibodies. Contractile proteins, I don't have space in the course for muscle activity, physiology, so actin and myosin in muscles. Storage proteins, particularly in eggs and milk, so you've got ovalbumin and casein as egg and milk proteins. And then toxins we've met. Oh, good old snake venom. That should ring a bell, for the time being anyway. So you can see that, you know, that really brings it home how much we've already met proteins. You know, out of that lot, you know, there we've had insulin, glucagon, hemoglobin, antibodies, snake venom in a roundabout sort of way. But the uses of them are, you know, so extensive. And the thing about proteins is that obviously the smallest protein you could have would be 2 amino acids. But the biggest protein you could have would be, well, you know, almost infinite, wouldn't it? And theoretically, there's an almost infinite number of possibilities, because if you've got 20 amino acids that you could use, you can put them together in whatever order you like. So let's say we've got a small protein which is 10 amino acids long. That is not big at all. How many different amino acid sequences could you have? This is challenging in maths. All right, you've got 10 amino acids. 3, 4, 5, 6, 7, 8, 9, 10. So? No, it's not times. How many could you have here? You could also have, so it's not adding them, because for each one here you could have 20 options there, so they're multiplied. So it's actually 20 times 20 times 20 to the power 10, which is fairly big. You know, if we were to work with something smaller, let's just take a 3. All right, there's a 3 amino acid peptide. So we've got 20 to the power 3. What would that be? Well, 20 to the power 2 is 400, and that to the power 2 is, sorry, to the power 3. Let's try that again. 20 times 20 times 20. So 20 times 20 is 400. 400 times 20 is 8,000. Yeah? So you can have 8,000 tripeptides. Now, you look at the size of those proteins that we just had on the slides. It would be hundreds of amino acids long. I mean, obviously, there are some sequences that won't work. There's a lot of sequences that won't work. But potentially, there are huge numbers of proteins to do different jobs. There is out there on the internet a protein data bank, or database, I'm not sure which it's called. I only met that this week because it is part of this module they're looking at Yarmouth to do with drug interactions, and you can actually look for interactions between drugs and proteins. But you know, part of the first stage of designing a drug would be to really find a target protein that's produced in the body, and then find molecules that will interact with it in certain ways. So proteins, you know, as I say, is a real biggie. Right, we will park the content at that point.